Good afternoon. Welcome back. We're going to talk about attachment and entry today. This is how virus particles get into cells in order to begin the reproduction cycle. So now we've finished a lot of the basics that you need to know to understand viruses. So now we're going to go through the reproduction cycle step by step for the next 9 or 10 sessions or so. So as you know, viruses have to get in cells in order to reproduce. They're obligate intracellular parasites. They can't reproduce outside of a cell. And they're too big to simply diffuse across the plasma membrane. Small molecules can do that, but viruses cannot. So they typically are brought into cells by pre-existing pathways that exist to bring large things into the cell. And that's what we'll talk about today. So the steps involved here are first, the virus particles, of course, have to adhere to the surface. So virus reproduction produces lots and lots of particles because they have to just randomly bump into cells when they get into a new host. So they're always bumping into cells. There are random collisions that occur, and there's electrostatic interactions that attract the particles to the surface, so there's no specificity there. If that cell doesn't have the right receptor for the virus, that is a molecule on the cell surface, the virus will move on most likely to another cell. So step one, random collisions, and then step two, attachment to a specific receptor. And sometimes there's more than one receptor. It can be a protein or more than one protein. It can be a carbohydrate, a lipid. We'll explore some of those today. And then after receptor interaction, the genome needs to get in the cell. And there are a number of ways that happens, which we will talk about today. Now, cellular receptors for viruses are essential, except, interestingly, for viruses of fungi. Fungi have viruses. Everything has a virus, right? But fungi have viruses, and those do not have to get into cells with a receptor. They don't have an extracellular phase, actually. They just reproduce inside the fungus, and then they go to the next fungus as the fungus divides. Same with yeast, a fungus. They have viruses in them. Some of them actually have capsids, and they still divide and assemble and move from cell to cell. And plant viruses don't need receptors. They get inside plant cells by some kind of mechanical damage. Could be an insect or a worm that is feeding on the plant and introduces viruses. Sometimes farmers inadvertently spread viruses. They walk through rows of plants, they get viruses on their clothes, and then they walk elsewhere and they bring them as well. So no receptors are known. So receptors for viruses as a field is pretty new. First one was identified, well, before 1985, but that was the only receptor that was known, which is sialic acid for influenza virus, as we'll talk about today. But after that year, many enabling technologies were developed, and they're listed here, including recombinant DNA, the ability to clone genes, monoclonal antibodies, an antibody against a single epitope, a single amino acid sequence, not a mixture as you would have in your serum. Flow cytometry, the ability to use antibodies to sort single cells with a particular protein on the surface. Nucleotide sequencing, of course, so you could sequence the genes. Small interfering RNA, so that you could knock down the levels of mRNAs and see if that is in fact an important receptor. And more recently, CRISPR-Cas9, where you can disrupt genes. So if you had a gene that you thought was a virus receptor, you could disrupt it by CRISPR-Cas9 to see if it is actually your receptor or not. So some of the criteria for identifying cell receptors, first, you want to show that the receptor binds your virus particle, of course. Thank you. Second, you might want to make an antibody against the receptor and show that that antibody blocks infection. So the antibody is directed against a protein on the cell surface, and that will block virus attachment. Third, if you can get the gene encoding the receptor and put it into a cell that does not have the receptor already, it confers susceptibility. Remember that word. Susceptibility means you have a receptor for the virus on the cell surface. So if you have cells that don't have the receptor here, and you add your gene in the form of DNA, those cells, some of those cells will now produce the receptor and they can be infected. It's a very powerful way to say, yeah, this gene encodes the receptor. Of course, if you need two genes, it makes it a little trickier. And finally, you disrupt the gene, the receptor gene, in a susceptible cell and show that they're not infected. So all of these are needed to show that a particular gene encodes a receptor or a protein that's a receptor for a virus. Now, again, very early on in the SARS-CoV-2 pandemic, ACE2, a cell protein, was shown to be the receptor for the virus. This was easy to figure out because other coronaviruses, including SARS-1, the original OG SARS, right? That one bound to ACE2. And so what was done here is to take HeLa cells, which don't make ACE2 normally, so that's good. And you may ask, how do you know that? Well, you could take an antibody to ACE2 and try to stain the cells and show that they're not stained, or you could look for transcripts of ACE2, which would be more sensitive, but say by PCR, but HeLa cells don't make ACE2. So you put in a plasmid encoding ACE2, which had been cloned years ago. You now have HeLa cells that produce ACE2. And so what do you do with those? Well, here is the experiment. You infect them with virus, and you see if the virus gets in. So let's see, here on the bottom, we'll start with HeLa cells alone. And we have four different panels here. We have a DAPI panel, which stains the nuclei, so you can see your cells. They're very faint here, but you can see the blue nuclei. And then in the second, we have an antibody to ACE2. And that, we use a second antibody that will be green under UV light. FITC is the green dye, is what fluoresces green. And then the third is an antibody against nucleoprotein, which is staining for virus. And then the last is emerge. So you can see if the cells that make ACE2 are the same ones that have the viral protein. So HeLa cells do not have ACE2. They don't get infected. So these are all infected. So there's no nucleoprotein staining. And now let's go back to the top. We have human ACE2 put into these cells. So in this experiment, they actually used human, bat, and mouse ACE2. And you can see the second panel, these cells are all producing ACE2, but only the human and the bat ACE2 allow the cells to be infected. And you can see by the merge that the cells that are infected are the ones that are making ACE2. Of course, not every cell is infected. And you could probably figure that out by knowing what multiplicity of infection they used here. Is the nucleoprotein antibody getting into the cell? This is a good question. Yes, because what you will do is permeabilize the cells with methanol before you add the antibodies. Otherwise, the antibodies can't get in. That's correct. So if you want to see cell surface proteins, you don't permeabilize. And this would tell you that ACE2 is on the cell surface. But for nucleoprotein, yes, you have to treat the cells with methanol, and then the antibodies will get in, okay? And NP is only in the cell. So the human ACE2 gives you susceptibility, so does bat, but not mouse ACE2. So this told them that the receptor is ACE2, and you could then go on and say these cells infected and so forth. But this gives you the ability to quickly identify the receptor. Now, having mice not have ACE2 or not be able to bind, murine ACE2 does not bind the virus, does not allow reproduction. And so that is unfortunate because it means that to make a mouse model, you have to do something else. And we'll talk about that later, what you have to do. Many receptors for viruses have been identified. These are all mainly plasma membrane proteins. So the plasma membrane is just shown at the top to remind you that it's a lipid bilayer with all sorts of proteins embedded in them, transmembrane proteins, indirectly anchored proteins, which means they're interacting with another transmembrane protein. There are proteins that are linked by lipids, lipid-anchored proteins, and so forth. There are lots of carbohydrates on these proteins as well. So on the bottom are some cell proteins that have been identified to be cell receptors. We have, for example, herpes simplex virus binds to heparan sulfate proteoglycan. We have influenza virus binding to sialic acid, which is a glycoprotein present on basically many proteins. We'll talk about that today. And then a variety of transmembrane proteins that serve as virus receptors. Let's see, I don't have any GPI-linked proteins. So some proteins are not attached with a transmembrane domain. There's a lipid that attaches them, but they're not shown here. Anyway, you can see poliovirus, herpes virus, foot and mouth disease. HIV requires 2 receptors for cell entry. We'll talk about that today. And so does adenovirus, requires 2 different proteins. So the HIV was hard to identify the receptor because 1 wasn't enough. They had CD4 early on, but it wouldn't confer susceptibility, and they had to look for another protein. That's where they came up with this multi-transmembrane, 1, 2, 3, 4, 5, 6, 7 transmembrane protein, which is a chemokine receptor. The main point of this slide is that all these proteins have cell functions, right? They don't exist so that viruses can get in, right? Some people think they do. None of you do, I'm sure. But some people out there in the world think that receptors exist for viruses. No, they don't. They have cell functions, and viruses have evolved to be able to bind to them. Sometimes different viruses can bind the same receptor, and there are 2 examples here. Adenovirus and Coxsackie virus B3 have a similar receptor. It's called the Coxsackie adenovirus receptor, or CAR. This is adenovirus on the left. Coxsackie virus is a picornavirus related to poliovirus. Coxsackie virus was isolated in Coxsackie, New York. Anybody from Coxsackie, New York, or nearby? No? Some years there are people. That's a town upstate, exit 21B. I took that picture holding my camera up as I drove by many years ago, because I always wanted a picture of it for my lectures, so I have it. And there was an outbreak of paralytic disease in the 40s there, and they thought it was polio, and then when they isolated virus, it was not. It was a different virus, and they called it Coxsackie virus. You know, nowadays, people don't want viruses named after their towns and cities. Did you know that? They really vigorously, that's why, well, I won't say it. Never mind. I don't want to get political. I'm not going to get political. But many, I'll give you examples throughout this course of that. But in the old days, they did, and we haven't changed them. And swine herpes virus, called pseudorabies virus, also binds the same receptor as poliovirus. So sometimes different viruses can bind the same receptor, or viruses of the same family can bind different receptors. So here, retroviruses bind 3 different rhinoviruses bind 3 receptors. We'll talk a little bit about that today. Retroviruses bind 16, and sometimes one virus can bind multiple receptors. Here's a herpes virus, herpes simplex 1, has 1, 2, 3, 4, 5, at least 5 glycoproteins in its membrane, and they all bind different receptors on the cell. And you may say, what's the function of that? What's the function of a virus binding to so many receptors? It gives it the ability to infect different tissues and cell types. And we'll see that when we talk about these viruses in a little more detail. All right, so that's an overview of virus receptors. Now let's talk about how virus particles attach to them. And here are 2 examples that I think illustrate one strategy, which is if you have viruses that are just a protein shell, remember icosahedral symmetry, and that's poliovirus and rhinovirus, they can interact directly with the receptor. So the receptor for poliovirus is a molecule called CD155, which actually we identified in my lab way back in the 90s, one of the first virus receptors to be identified. We used to call it the poliovirus receptor, which I liked because the abbreviation was PVR, Vincent Racaniello, you know? I like that. But some committee renamed it. There you go. Anyway, this receptor binds into a groove around the 5-fold axis of the virus. In this little groove, it's called the canyon. And many years ago, we solved the structure of that receptor bound to the virus particle, and it's shown here. So we added saturating amounts of receptor. So every receptor binding site was occupied. And there's 60, because that's what you would predict from icosahedral symmetry. There should be 60 copies of everything. And there's 60 copies of receptor bound to the virus particle surrounding the five-fold axis, 1, 2, 3, 4, 5. So that's one approach, is that the receptor can bind in a groove. I have to show you this. It's on my phone. See? This is the poliovirus receptor complex. I don't have my kids on my phone. All my pictures are of viruses, no kids, okay? Because I love viruses. Anyway, I also have other polioviruses, which I'll show you when we get to other parts of the course. On the right is rhinovirus bound to its receptor. Rhinovirus has a canyon, just like polio, this little groove around the five-fold axis, but the receptor doesn't bind there. The receptor is shown in gray, just as for polio. And here, the receptor is low-density lipoprotein receptor, LDLR2, I think. And you can see it sits on the top of this, at the five-fold axis. It's a flat molecule sitting right on that top. It's called a plateau. It's on this diagram here. It's just this little plateau at the five-fold axis. So the LDL receptor fits there. So the moral of the story is, just because there's a lock doesn't mean a key fits into it, okay? So don't be fooled. If there's a canyon, that may not be the receptor binding site. So very different. But the point is that the receptors can interact with protein directly. For adenovirus, it is the fiber that binds to the receptor. So here's the icosahedral adenovirus we talked about last time. And at each five-fold axis, there is a fiber protein coming out with a knob at the end. And here in the middle is the structure of that fiber, made up of a shaft, which is basically three proteins, same proteins twisted around each other. And then at the end, they form a knob. And the knob sits down on the receptor. So there are two receptors for adenovirus. There is a fiber receptor. So this is the fiber. So there's a receptor for fiber shown there at the right. And then there's a second receptor, an integrin receptor, that's needed to bring the virus into the cell by endocytosis. We'll have a look at that later. So the knob sits down on the fiber receptor, which is called CAR, Coxsackie adenovirus receptor, because it's shared with Coxsackie. And this is a top-down view of CAR in blue, and the knob just sitting down on top of it. So that's a very striking example of a protein receptor interaction. Now, for membrane-containing viruses, it's a little bit different. They attach to receptors via their spike glycoproteins that are stuck in the membrane. So here on the left, influenza virus is an enveloped virus, and different spike proteins are embedded in the membrane. We're going to encounter these a lot in this course. So hemagglutinin is one of them. Then there's the second neuraminidase. But hemagglutinin is the one that attaches to the receptor. So here in the middle is the X-ray crystal structure of the hemagglutinin. It's a membrane-bound protein. It has an extended fibrous stem. It's perpendicular to the membrane. And then at the top is a globular head, and inside of it fits the receptor. So if we look top down into that globular head, we have the picture shown here in gray on the right. And there's the receptor for the virus bound in it. It's sialic acid, which is a small sugar. I'm going to show you the structure of that in a moment. It's a sugar that's typically attached to glycoproteins, and so the virus binds to that sugar, and the sugar can be on any protein pretty much. Right? So sialic acid is the receptor. And so here is sialic acid. There are two kinds of sialic acid shown here on the top and the bottom. Sialic acid is the moiety on the left here, and then on the right is the second sugar to which sialic acid is attached. So this is typically part of a oligosaccharide. So here on the left is a model transmembrane protein. And the extracellular domains typically have chains of sugars attached to them. We call those oligosaccharides. And so sialic acid is always the last sugar on a chain. It's the yellow one there. And so that's the yellow structure here, sialic acid. And then on the right, it can be a variety of sugars. We're just showing galactose as an example. And so sialic acid has a 6-carbon ring with various side groups attached to it. And there are two kinds of sialic acids that are receptors for influenza viruses. So again, this is the moiety that fits into the hemagglutinin that I just showed you. There are two shown here, depending on how the sialic acid is linked to the second sugar. It's either an alpha-2,3 or an alpha-2,6 linkage. All that means is that the number 2 carbon of sialic acid is linked via an oxygen to either the 3-carbon of the next sugar or the 6-carbon of the next sugar. Very simple nomenclature. So influenza viruses that infect humans preferentially bind to alpha-2,6 sialic acids. That's why I have the people here to remind me that. And influenza viruses that infect birds preferentially bind to alpha-2,3. And birds are the main reservoir of influenza viruses in nature. Whenever I have a new influenza pandemic, part of the virus is usually coming from birds. We'll talk about that later. All right, so those are the receptors for influenza. So there are actually 3 kinds of influenza virus, A, B, and C. So A, B, and C bind this kind of sialic acid, which is, the chemical name is N-acetylneuramidic acid, because there's an acetyl group attached to this nitrogen here. And then there's another kind of sialic acid, which would be 9-O-acetyl, where the acetyl group is attached to an oxygen, number 9, and those are the receptors for influenza C viruses. HIV is another example where attachment's been highly characterized. Here's the particle on the left. Now notice, I don't have a lot of glycoproteins here. Now the artist who drew this didn't just do this on his own. This is actually how it is compared with influenza, where the glycoproteins are packed very densely. And they're not in HIV, and that makes it hard to make vaccines against HIV, because if antibodies can't cross-link the glycoproteins, it's hard to block infection. We'll talk about that later. So there's an envelope glycoprotein for HIV. It's a trimer shown here in the upper left. Here's the structure of it shown here, and the receptor for this glycoprotein is CD4. Here's CD4 in purple here, and the gray molecule is part of the trimer glycoprotein that's binding CD4. CD4 is, of course, the protein that defines CD4 T cells, right? It's the marker that was originally used to define those. And it is a receptor for HIV. Here is a blow up of this internal region here to show you that there is a phenylalanine at this part of CD4, which is sticking into a pocket in the glycoprotein of HIV. And this is essential for the interaction. If you change that phenylalanine, the proteins don't interact with each other. So very specific interaction. Of course, our latest addition to virus receptor interactions is SARS-CoV-2. SARS viruses, coronaviruses are also enveloped. They have a spike glycoprotein in the membrane. And this, for SARS-CoV-2, I've shown you, of course, that the receptor is ACE2. And so here is the structure of the spike glycoprotein of SARS-CoV-2. It is a trimer. This is just a monomer shown here. So there are 3 of these that are involved in attachment. And the different domains of the protein are colored. And the green one is the receptor binding domain. That's the part that binds ACE2. And in fact, on the right is the structure of the spike bound to ACE2. ACE2 is in red. It's binding the RBD. RBD is really important because most of the antibodies that block infection bind the RBD, hence they're interfering with the virus receptor interaction. Now, other coronaviruses, their receptors have been identified, and those are just shown here. So you see that ACE2 is also bound by SARS-CoV-1, as well as NL63. So this is a coronavirus that causes common colds. There are 4 of them, only 3 shown here. They've been known for years, just cause mild infections. But the receptors for those are known. You can see some of them are different. And we have one that uses N-acetylneuramidic acid. It's the same receptor as influenza C virus. Viral receptors on the cell surface, A, can bind directly to icosahedral virus capsid proteins. B, interact with glycoproteins of envelope viruses. C, can be carbohydrate or protein molecules. D, have cellular functions. E, all of the above. All of the above, that's correct. Most of you got that. So these receptors can bind directly to icosahedral particles. I showed you examples of polio and rhino. They interact with glycoproteins. We have HIV, we had influenza, SARS-CoV-2 as examples. And none of you picked the other two, which is good because it's all four. They can be carbohydrate or proteins, and they have cellular functions. All right, so we've bound a virus particle. How do we get it into cells? Cells take up things routinely. They need to take things up, and there are a number of mechanisms for that shown on this slide. So we have phagocytosis, where large particles tend to be brought up by cells. And then we have endocytosis in the middle, where smaller molecules are taken up into cells. And the way endocytosis can work is you have typically pieces of the plasma membrane coming out, forming these little extensions. And here we're showing it grabbing small molecules and taking those into the cell. The small molecules are taken into an invagination. The invagination pinches off and becomes a vesicle. So now these molecules are in the cell. So that's called macropinocytosis. I think pinocytosis means cellular drinking, something like that. And on the right is receptor-mediated endocytosis, where there is a receptor for some ligand on the cell surface, and the ligand binds the receptor, and then the complex is taken up into the cell by endocytosis. So the process on the left, the macropinocytosis, is not specific. There's no receptor involved. But on the right, it's the specificity is conferred by the receptor. So for example, our cells have ferritin receptors on their surface for the protein ferritin, which is bound to iron. And so to get iron in the cell, the iron is bound to a protein, and the protein binds a receptor, and then that's taken up into the cell. And so viruses are brought in by these two central pathways here, macropinocytosis and receptor-mediated endocytosis. On the right, there's another mechanism of entry called filipodial capture. Cells make these projections by virtue of pushing actin out, and virus particles can bind them and move down and eventually get into the cell. So the cell is very crowded, so things don't just diffuse. Everything that moves around within a cell does so by some kind of transport mechanism. Here's a drawing of a cell by David Goodsell, who's a wonderful biological artist. And I've broken it into pieces because they're too long to put on one slide together. So here is the plasma membrane with proteins in green. And then we have the actin microfilaments below it. And then if you keep going in, you see some ribosomes in here and microtubules. And then we move further in. Here's some Golgi and endoplasmic reticulum. Moving further in, we encounter the nuclear pore. This is the nuclear membrane right there. And there's a nuclear pore, and there are proteins being transported in. And then we enter the nucleus, of course, which is full of DNA wrapped around nucleosomes. So it's very crowded, and things are not going to just diffuse. In fact, people have calculated that it would take hours for a virus to diffuse to where it needs to go in the cell in order to reproduce. And so there's always some kind of active mechanism. And here's a summary of all these different mechanisms that lead to virus uptake. Of course, this is just one cell showing them all together. Viruses that have envelopes, those membranes have to fuse with the cell membrane at some point. And that fusion can occur at the plasma membrane, as shown here, and then you would have the nucleocapsid released. In this case, that nucleocapsid is containing a DNA, so it needs to put the DNA in the nucleus. So the nucleocapsid is transported on a microtubule by motor proteins to the nucleus. So not diffusion, always some kind of transport. Here's an example of an icosahedral virus, adenovirus, being taken up by endocytosis, and again, transported down microtubules by motor proteins to the nucleus. Here is an example of endocytosis, in this case of an RNA virus. The virus particle is taken up into a vesicle. The vesicle pinches off and moves down, and as it moves to the nucleus, it's moved by motor proteins on microtubules. Same for influenza viruses and polyoma viruses as well. So the point here is that entry can occur from the cell surface. It can occur from within an endosome, and it's always moving around by motor protein. So here's an example of such a motor protein. This is a scientific animation by ex vivo. That is an endocytic vesicle, and the thing that's walking, looks a little bit human, is a motor protein. which is attached to the vesicle, and it's attached to a microtubule, which is the filament below here, and this is the cytoplasm as well. So a virus particle could be in that, and it's being brought down from the plasma membrane into the nucleus. Okay. So let's talk about entry at the plasma membrane. So many viruses do this. Here's a summary at the top. You have your virus particle, envelope virus particle with glycoproteins attaching to a receptor. Many viruses fuse right at the plasma membrane and release the genome right there. So they don't need to get into the cell by endocytosis. And how does that happen? It has to be regulated because viruses can't fuse when they bump into any old membranes, right? Because then viruses, as I told you, are always bumping into cells. And if they fused with the first cell they bumped into, it would be a lot of non-productive events. So there's always specificity. And I'm going to tell you how that works here for two viruses. First, the paramyxovirus, like measles virus. The virus is shown at the top, and the virus has two glycoproteins in it. It has an attachment protein and a fusion protein. The attachment protein binds to a cell receptor. So the attachment protein shown in green, cell receptors shown in purple. When the attachment protein binds to the receptor on the cell surface, it induces a conformational change in the attachment protein, in the viral attachment protein. And that in turn causes a change in the fusion protein, which is right next to it, such that the fusion peptide now inserts into the cell membrane. So the fusion peptide has at its N-terminus a very hydrophobic sequence. Hydrophobic amino acids can insert into membranes. But you can see in this slide, they're hidden. And in fact, not only are they hidden, but they're internal. The little jagged piece there, that's the fusion peptide. It's internal, and so it requires a proteolytic cleavage event for it to be exposed, and it also requires this conformational change to be inserted in the membrane. And once the fusion peptide inserts into the membrane, then the two membranes can fuse by mechanisms we'll talk about in a moment. So in that case, attachment to a receptor and cleavage regulate fusion. If the cell doesn't have the right receptor, virus won't enter. On the bottom is another strategy shown by HIV where we need two proteins to get fusion. So fusion is regulated by two proteins. So the HIV particle has an attachment protein that has two components. It has an attachment portion called GP120, and it has a fusion peptide as well. The fusion peptide is also hidden. It's bent up towards the virus membrane. What happens here is that GP120 attaches first to CD4, which is the first receptor. CD4 attachment induces a conformational change in GP120 such that it can now bind the second receptor, which is a chemokine receptor, CCR. And then finally, that second interaction causes a conformational change in the fusion peptide or the fusion protein. It flips out the peptide into the membrane. All right, so two receptors are needed in order to flip out that fusion peptide. And once the fusion peptide's in the membrane, it can catalyze fusion. So here is a movie of HIV-mediated fusion. So the fusion protein is extended. Here's the viral membrane down here, and the fusion peptide is now stuck in the cell membrane. This is a trimer, remember. So we've already bound the receptors. The fusion peptide has gone in to the cell membrane. And what is going to happen now is these proteins are going to reorient. You can see this protein here is folding back, and that's drawing the two membranes together. And when membranes get close and you get rid of all the water molecules, they will fuse. That's what you need to do, actually, is get them so close to exclude water. That's energetically difficult to do, but these two proteins doing those motions do that. Now, the reason those proteins do that, they're extended. They don't like to be extended. They like to be in a hairpin conformation. So they spontaneously fold back on each other, and they bring the membranes together. And when you bring membranes together, you can form a pore, so the genome can now go out of the pore. All right, so that's how fusion for HIV works. You have initially an extended fusion peptide. It hairpins back to bring the membranes together, and you have a pore forming. We'll see some other examples of this as well with other viruses, in case you don't quite get that. All right, the next question, which of the following does not play a role in virus entry? Clathrin-mediated endocytosis. It's a kind of endocytosis. I didn't mention clathrin, but clathrin is a certain kind of endocytosis. Fusion of viral and plasma membranes, diffusion of virus particles in the cytoplasm, microtubule-mediated transport, sialic acid. So which does not play a role in virus entry? Not which does, but which does not. Diffusion is not what's happening. That's right. All the others are correct. Clathrin-mediated endocytosis happens. Fusion of viral and plasma membranes happens. Microtubule-mediated transport, that's the movie I showed you, sialic acid. So it's diffusion that does not occur. So let's look at fusion for influenza virus. It's been well studied and illustrates a lot of the similar things as for HIV. But I want to focus on this because it'll be important for later on. So the virus binds to the cell receptor. So HA on the virus binds to sialic acid. So that's the first panel there. The virus is being engulfed in what's going to be a vesicle at the cell surface. And on the bottom here, we have a detailed picture, a close-up picture. We have two hemagglutinin molecules. So the globular head there is binding to sialic acid. We don't show it in this picture. Remember, the HA is a trimer. The globular head is binding to sialic acid. Now, this little invagination pinches off and becomes an endosome. And endosomes move towards the nucleus by microtubule transport. And as they move to the nucleus, the pH drops. This is a normal part of endocytosis. The pH drops to between 5 and 6 from 7. How the pH drops is that protons are pumped in to the endosome by a pump in the endosome membrane. So they get pumped in because the cell wants to acidify endosomes because often ligands in the receptors can be separated at lower pH. So the transferrin can be separated from transferrin receptor at low pH, okay? So the pH drops, and as the pH drops, that causes a conformational change in the hemagglutinin molecule. So you can see now here, the heads of the globular heads are falling away as the pH drops, and eventually they're all the way away, and the HA, the rest of the HA, extends so that the fusion peptides are inserted into the endosome membrane. So that's on the third panel here, those orange tubes, those are the fusion peptides stuck in the cell membrane. Normally, those at neutral pH, those are hidden. You might be able to see the little red jagged lines right near the viral membrane here. Those are the fusion peptides. So the HA is folded back on itself to hide those fusion peptides. And as the pH drops, the HA extends and the fusion peptides go in the membrane. So that's the function of the drop in the pH, to extend the HA. But the HA does not like being in an extended conformation. It's not energetically favorable. So it starts to hairpin back, just like as we saw for HIV, and that's shown in the fourth panel here. The HA chain is folding back on itself, and that of course draws the membranes together, and eventually the membranes will fuse. You have now a pore through which the viral genome can go. You can see now the HA is folded back on itself. So this is called hairpinning because in the folded conformation, it kind of looks like a hairpin, right? And then in the extended conformation, you've taken that hairpin and just bent it open. And you have to imagine that the hairpin would want to fold back on itself. I don't think a real hairpin would do that. I think it would be bent permanently, but biologically, the HA doesn't like to be extended, so it folds back and that causes the fusion. Really, really elegant mechanism. And eventually, the RNAs of the virus come out through that fusion pore, and they will eventually go in the nucleus, and we'll talk about that process another time. So that's HA-mediated fusion. Now, the fusion of HA is regulated by a cleavage event, a proteolytic cleavage event. So here is the HA, the native HA. There's a cleavage site down here near the viral membrane. The red is the fusion peptide. And in order for it to be exposed when the HA undergoes this conformational change, the fusion peptide has to be cleaved. And that little Pac-Man thingy there, that is a protease that is cleaving the HA. And so the middle panel shows you the cleavage has happened. So now the N-terminus of the fusion peptide is available. And then when pH drops, you can now extend the HA, and the fusion peptide is all the way at the top there. So remember, it goes from being near the bottom, near the viral membrane, gets thrust up into the cellular membrane. So that's the extended hairpin, or the bent hairpin, and then it hairpins back to fold back and bring the two membranes together. So the cleavage is absolutely essential for the fusion peptide. If this weren't cleaved, this extension could not occur. If you look here, these two alpha helices, the blue and the brown one, see the brown one ends up on top. So the whole protein opens, thrusting the fusion peptide up. If it weren't cleaved, that would not happen. So what cleaves it? Proteases that are mainly present in the respiratory tract, which is why influenza virus is mainly a respiratory virus. So there are a lot, these are called class 1 fusion proteins. There are a lot of them for different viruses. Influenza virus we have talked about here. These are all perpendicular to the membrane. They're mostly alpha helical. All these extended conformations are alpha helices, and the trimers. So we have influenza virus, simian virus 5, Ebola virus, HIV, retrovirus of mice. They're all type 1 or class 1 fusion proteins with these characteristics. Here is a movie of influenza virus entry, though the virus is bumping into a cell. It's binding to sialic acid. It's being taken up by endocytosis, and the endosome is coated with clathrin, and so they're clathrin-coated endosomes. And it's moving, it's going to find a microtubule at some point and there you go, being transported into the cell. So the artist knew not to let it diffuse. That's good. Clathrin comes off, which is a normal process. So inside that, the pH is dropping. And now we have fusion of the virus and cell membranes, and out come the viral nucleocapsids. Those yellow things are viral nucleocapsid. They're going to go into the nucleus by the pore. So you don't have any details there, but you saw the fusion of the virus to pop out of that endosome. How about SARS-CoV-2? So this has been extensively studied, obviously, as well. There's the spike protein bound to ACE2. ACE2 is in red. There's a fusion peptide for the spike protein here as well. It is this little cyan-colored alpha helix about halfway down the spike. And that fusion peptide, if you look at a linear representation of the spike, it's 1,273 amino acids. That's why the mRNA vaccine was called mRNA 1273. And we have the receptor binding domain there in green. The fusion peptide, you can see, is in the middle of the protein. And so we need cleavage of this protein so that the fusion peptide eventually will be exposed. And this protein, very much like influenza virus, undergoes a conformational change to put the fusion peptide near the top there on the right, that cyan-colored part. But this does not require low pH. Right? This is a differently mediated, this fusion is mediated just by cleavage of the spike protein of SARS-CoV-2. So it needs to be cleaved, and in fact, there are two cleavage sites, S1,2 and S2', and you can see that S2' cleavage site will then allow the fusion peptide to be liberated and inserted into the membrane when the protein undergoes its conformational change. So what causes the conformational change of SARS-CoV-2? And here, in explaining this to you, I'm going to tell you why hydroxychloroquine could never work for COVID, okay? Despite, to this day, do you know there are physicians who prescribe hydroxychloroquine? It cannot work for the following reason. In the respiratory tract, there is a cell surface protease called TMPRSS2, TMPRSS2. And when SARS-CoV-2 binds ACE2 on the surface of respiratory cells. TMPRSS2 cleaves the spike at the S2' position. Upon cleavage, the protein reorients and catalyzes fusion. So fusion occurs at the plasma membrane. Doesn't need endocytosis, all right? There is an inhibitor of TMPRSS2 that was considered for therapeutic use. It's called Camostat, but it never was used because after the hydroxychloroquine debacle, people got scared of targeting this part of the pathway. So what about hydroxychloroquine? So hydroxychloroquine's antiviral properties were studied initially in a cell line called Vero cells. These are vervet green monkey kidney cells that are used in labs. They grow lots of viruses. You can grow coronaviruses in them. And the problem with Vero cells is they don't have cell surface TMPRSS2. So the virus will bind ACE2 on those cells. It'll get taken up by endocytosis. The virus is delivered to the endosome, and there's a protease called cathepsin L that will cleave the spike and it will trigger fusion. So the virus gets its genome out of the endosome. It is cathepsin L that's inhibited by hydroxychloroquine. Your lung cells don't allow entry this way. The virus enters at the cell surface. So hydroxychloroquine does not inhibit cell surface entry in lung cells. The fault here was that the people who used Vero cells were not thinking, they were not virologists, and they didn't realize that these cells are biologically different from lung cells. So they published a paper saying hydroxychloroquine inhibits reproduction in Vero cells. And everyone went nuts. And that's why we got to the place we are today. But if you just look at the biology, you know that there's no way that hydroxychloroquine can work in lungs because the virus fuses at the cell surface. So I like how basic science can explain why things do and do not work. Now we need to get our politicians to know some basic science. So maybe one of you will be president of the US someday and you'll know this. That would be great. Okay, just send me an email and thank me for teaching you stuff. There are also class 2 fusion proteins. They are parallel to the membrane. We've seen these already on some flaviviruses. They're mostly beta sheet as opposed to the alpha helices. They make dimers. So here's a flavivirus, Zika virus, where these are laying parallel to the membrane. But the fusion still works in a similar way. So here we have one of these class 2 fusion proteins parallel. There is a fusion peptide which is hidden in this assembly here. So this is the structure. The fusion peptide is hidden in the interface between the proteins. Upon low pH, these proteins reorient. They put the fusion peptide in the cell membrane. They then hairpin to form a fusion peptide. And so they can work in very much similar ways as class 1 fusion proteins. And here's a movie of dengue virus, a flavivirus, which has a class 2 fusion glycoprotein. You can see they were laying flat on the surface in that initial view. And now the endosome is coming in. We're looking inside. And as the pH drops, the glycoproteins are reorienting. The fusion peptides are going up. And they're going to be interacting. They're going to be inserting, of course, into the cell membrane. You can see that there. And then they're going to undergo hairpinning to bring the viral membrane, which is at the bottom, and the cell membrane, which is at the top, close enough together in order to fuse. And there's the fusion pore, and the nucleic acid of the virus is about to come out. There it is, and leave the endosome and go out into the cytoplasm. This is a plus-stranded RNA virus, so that RNA is going to be directly translated. So Ebola virus is interesting because the receptor that catalyzes fusion is actually within the endosome. So these are very long filamentous viruses. They're probably taken up by some kind of macropinocytosis. Nobody's ever identified a specific cell surface receptor. So I think they're big enough that they can be just non-specifically taken up into cells. They get into an endosome. In the endosome, there's those cathepsins that I just mentioned for SARS-CoV-2. They cleave the glycoprotein and expose a part of the glycoprotein that will then interact with a endosome receptor called NPC1. And that interaction leads to fusion of the viral and endosome membranes, and the genome is released. NPC1 is a cell protein that's involved in cholesterol transport. And there are some people who are defective in NPC1, and they unfortunately die in their teens. They have real problems because cholesterol isn't moved around properly in cells. And part of the demonstration that NPC1 is an Ebola virus receptor was to take fibroblasts from those patients and show that they're resistant to Ebola virus infection in culture. So they would never be infected by Ebola virus, but unfortunately they don't live very long to see that benefit. So one of the key takeaways that I want you to get from all this is that fusion has to be regulated. It can't happen in the wrong place, because if viruses are bumping around into membranes all the time, they would fuse non-productively. So how do you regulate fusion? Well, at the plasma membrane, where there's no pH drop, as we see in endosomes, you need a second receptor interaction. So here it's shown for HIV, which is using a second receptor. And in the endosome, we depend on low pH to catalyze fusion. So that's not something you would normally encounter outside of the cell. And you also depend on cleavage of the spike protein to activate it for fusion. So if the SARS-CoV-2 spike is not cleaved, it is not going to be able to fuse to cells. Same for the influenza HA. And for some viruses, another regulation step has evolved, and that's to have the fusion receptor in the endosome. And so that prevents Ebola virus from just bumping into and fusing with any cell as it's moving around. Fourth question, viral fusion peptides are exposed for insertion into the host cell membrane when A, the virus particle is near a cell, B, the virus particle is in the cytoplasm, C, trimers of the fusion peptides form, D, the endosome becomes acidified, or E, the virus is docked on the nuclear pore. So fusion peptides are exposed when the endosome becomes acidified. But many of you picked other answers. So the virus particle is near a cell. That's not enough to get exposure of the fusion peptide, right? It would have to be attached to a receptor. The spike would have to be cleaved and so forth. Virus particles in the cytoplasm, well, by then it's already happened, right? When it's in the cytoplasm, the fusion has happened. Trimers of the fusion peptides form. A trimer per se is not a consequence of it's not needed for exposure of the fusion peptide. The acidification is what is absolutely required. So you can actually get this to happen without a trimer. I didn't tell you that, but D is really the best answer, and that's what these are all about. And when the virus is docked on the pore, no, because then, again, that's a long time after the fusion event has already happened. Finally, let's look at how a genome gets out of a non-envelope virus. Envelopes are easy because they just have to fuse with some membrane. What about like adenovirus or poliovirus? So here's adenovirus binding to its fiber receptor initially. Then it needs to bind with an integrin in order to be taken up by endocytosis. And then as the virus moves into the endocytic pathway, the fibers are released. Those come off very early on. And then as acidification occurs, the virus particle begins to be disassembled by the low pH. And those little diamond proteins that are coming off the particle, so normally they're embedded into the icosahedral particle. When the pH drops, they're coming off, and those make a pore in the endosome membrane. They actually poke a hole in the endosome membrane. You can see that here. A few of those, again, diamond-shaped yellow proteins come out of the particle at low pH. They make a hole, and the particle then comes out. It moves by microtubule transport, and it docks onto the nuclear pore complex. So that particle is already partially disassembled, and then the DNA comes out through the pore into the nucleus. And here are two electron micrographs showing you Here, the adenoparticle, it's already come out of the endosome. It's on the microtubule going towards the nucleus. And here are two particles docked onto nuclear pores. So here's cytosol here on the right half and the nucleus on the left. And those are nuclear pores where the virus particle is docked. Now, at the nuclear pore, a very interesting event happens to further disassemble the virus. So on the left, we see the virus being transported on the microtubule by a dynein motor, docks on the nuclear pore. And you know, these proteins, these dyneins and kinesins, these are proteins that attach cargo to the microtubule, right? There's also attached to the capsid a kinesin, and the kinesin pulls the capsid the other way. So dynein motors bring things from the plasma membrane to the nucleus, kinesins bring them out. And so adenovirus is sitting on the pore. The idea is that the kinesin starts to pull the capsid away, but it's really stuck on the pore and it breaks up, and that allows the genome to come out and get into the nucleus. So the opposing force of the motor pulling the capsid further breaks it up, the DNA comes out. And the pore, by the way, you need to have a nuclear localization signal on a protein in order to get in the nucleus. There's an import machinery that's involved in importing proteins. And so the DNA has to be linked to some protein. Nucleic acid typically will not get in without a protein that has a nuclear localization sequence on it. And so that's, in fact, this protein 7, which is bound to the viral DNA. It has. Here's what poliovirus does. Poliovirus binds its receptor, and the receptor itself catalyzes a conformational change in the particle and leads to the formation of a pore in the virus particle, which is shown here, is the 5 and the 3-fold axes of symmetry. So it's in this triangle between them. And this is on the right, a cryo-EM reconstruction of this pore. It's called the umbilicus connector. So here's the virus, and the endosome membrane would be just at the base of this umbilicus. So somehow the receptor causes a pore to be made through which the RNA then can come out, and it gets it across the membrane by virtue of this pore. And I had a little model for you of this on my desk, and I left it, but it's maybe I'll bring it next time. It's a lovely model of adenovirus showing how that might work. And there's another naked icosahedral RNA virus whose encoding is being sorted out, and that's a calicivirus. So caliciviruses include noroviruses, the agents of gastroenteritis. And for these viruses, when they attach to their receptor, a portal forms at one axis of the particle. So here's the virus particle here, and this is an image where a portal has formed. So when the virus docks onto its receptor, these are molecules of one of the viral proteins that reorient themselves, form a portal, insert into the membrane, and allow the nucleic acid to come out. Now, what's interesting here is for polio and this calicivirus, only one portal is made. So it's not like herpes virus where there's a preformed portal on the capsid. For these, there's no preformed portal, yet only one portal occurs. And the RNA has to come out by one end. So the end needs to be by a 5-fold axis. So the question is, how is it known where the RNA is to put the portal in the right place, right? I don't think it's known. I think it's random, and that's why some particles are not infectious. Remember the particle to PFU ratio. Not every particle is infectious. And it may be that, so you have 12 5-fold axes where a portal could form, and so you have a 1 in 12 chance of hitting that where the RNA is, or maybe even worse than that, depending on whether the end of the RNA is in the wrong place. But so that's the problem here. Where the RNA end is, the portal has to be near it. Reovirus are the viruses with 2 icosahedral shells I told you about last time. And now I'm going to tell you the function of those 2 shells. So these viruses bind receptors. They're taken up into the cell by endocytosis. The pH drops. And this virus is quite interesting because, in the normal endocytic pathway, the endosomes move in and they fuse with lysosomes. Lysosomes contain enzymes of all sorts, proteases, lipases, nucleases. And most viruses get out of the endocytic pathway before the lysosome fuses with the endosome. Otherwise, they'd be digested. But reoviruses and rotaviruses stay in the pathway, and they let the proteases remove the outer shell. So they use the proteases in the lysosome when it fuses with the endosome to take away the outer shell, and then the inner shell that's made has very hydrophobic proteins exposed, and those cause the particle to fuse with the membrane of the endosome and get out into the cytoplasm. So the fusion peptides, if you will, are beneath that outer shell. The outer shell is removed by lysosomal proteases, and then the virus can get out of the endosome. And so that's the function for having two shells. The outer shell is to protect the fusion peptide until the right condition is met, the proteases to remove it. So this is a summary of everything I've told you. Again, we saw this before. We can fuse at the cell surface. We can go through various types of endocytic pathways. We have clathrin-dependent endosomes. We have clathrin-independent. We have caveolin-dependent. These are all protein markers that mark these different kinds of endosomes. But the point is, the viruses can enter via changes in pH in the endocytic pathway, in some cases by enzymes in the lysosome. And all of these vesicles are brought throughout the cell by motor proteins on microtubules. They don't just diffuse. And one last word on nuclear entry. The nuclear pore restricts the size of things that can get into the nucleus. Most viruses are too big to just pass through the pore. The exception is the influenza virus genome, where it is actually small enough to be transported through the pore. And so that's how that happens. The genome is released in the cytoplasm as the fusion event occurs, and then the RNPs, the nucleocapsids, can go through the pore. The next panel there is herpes virus, where the particle docks on the pore with the portal docked onto the pore. You can see the portal there. And a good question is, how is it known to put the portal on the pore? We don't know how that happens. But then once the portal is there, the DNA can come out. Adenovirus is in the third panel, where I've shown you it gets disassembled on the pore, and then the DNA goes through it. And then the last example, there is a small virus, the parvoviruses, which actually could fit through the pore, but they don't. They bind to the pore and they cause disruption in the nuclear membrane and then get in that way. So these are just 4 different ways that viruses can get into cells. The last story I want to tell you is about how differences in the sequence of receptors can control infection. And so this is a story with rhinoviruses. These are icosahedral viruses without a membrane. They're classified into 3 types, A, B, and C. And there are 3 different kinds of receptors that are utilized by these, that are bound by these viruses. So the A and B types bind ICAM1, which is an Ig-like protein shown on the right there. The B, they also bind, 10 serotypes of these A and B also bind low-density lipoprotein receptor, which I showed you, bound to the particle already. The particle on the left has ICAM1 bound to it. And then there's also rhinoviruses type C, which bind a 3rd receptor called CDHR3. And CDHR3 is a curved molecule, membrane-bound molecule that attaches to the cell receptor. So the story I want to tell you is that there is a single amino acid change in CDHR3, which is a change to tyrosine, which causes increased surface production of the receptor. So the other allele that's present there is cysteine. You have lower cell surface receptor. So CDHR3, 529Y is a risk factor for kids who have wheezing illnesses. It's a risk factor for hospitalization for childhood asthma. So this was noticed a while ago, that kids who have asthma and go to the hospital, they tend to have, in this protein, 529Y. So position 529 can be a Y or a C, and the Y is associated with higher surface levels in asthma. Cells with a Y, with CCHR3 with a Y, 529Y, have higher surface levels. They also make 10 times more HRVC when you infect them. So having more surface protein leads to more infection and more virus production. And so what we think is the risk factor of 529Y is actually because those kids get more Rhino C infections when they're young and they're predisposed to getting asthma. All right, so that's the relationship between this Y allele at 529 and being infected with Rhino C. So humans don't all have 529Y. Many of us have 529C. And in fact, we think if you look in the genomes of Neanderthals and Denisovans, they have only 529Y encoding genes. They don't have 529C. So Neanderthals and Denisovans have 529Y, which gives you more severe rhinovirus infection. Humans have 529C. So what happened there is probably the virus selected for humans with 529C because it protects them against severe rhinovirus disease. So Neanderthals and Denisovans have Y. Probably the virus wasn't circulating back then. Otherwise, there would have been pressure for it to select the C allele. Okay, so that's very interesting in itself that we can look at the genomes of these ancient humans and see that. Finally, in 2013, there was an outbreak of HRVC in chimpanzees in Uganda. Very severe outbreak. A lot of them died. And how this happened was people visiting the place where they were kept gave them Rhino C. The humans had brought Rhino C, and the chimps all are homozygous for 529Y. So they got severe rhinovirus disease. And why are they homozygous? Because chimps don't get Rhino C unless they encounter humans. So living in the forests, there's no selection for chimps to make them have 529C. And so when virus is introduced to them, they get severe disease. All right, next time we're going to talk about RNA-directed RNA synthesis.